Our presentation is called A3 Cross in Drosophila melanogaster. Group members will be speaking in the order that they are listed. The objective of this experiment was to determine which Drosophila melanogaster chromosome the PW transgene was placed on. Here is some background information for better comprehension of the experiment. What is a transgene? A transgene is a gene that is placed at a locus on a chromosome. What is segregation? Mendel's law of segregation states that allele pairs separate or segregate during gamete formation, or meiosis, and unite randomly at fertilization. What is independent assortment? Mendel's law of independent assortment states that the separation of different pairs of homologous chromosomes into gametes happens independently of one another. This should offer a predictable ratio, for example, a 9:3 to 3:1 ratio. Fruit flies are named after their mutant alleles and not their wild type or natural alleles. An example of this is the dominant stubble bristle trait. The mutant allele is written capital S lowercase b and the wild type allele is written capital S lowercase b with a plus sign following. Now we will go through the crosses conducted in this experiment. Our experiment ID number was A3. Our parent flies consisted of a true breeding wild type female which had pigmented eyes, straight wings, and long bristles, and a true breeding mutant male which had white eyes, curly wings, and stubble bristles. These parent flies produced the F1 offspring that we sorted and collected for experiment 1. Of the F1 generation produced from the parental generation, we collected male flies with wild type or pigmented eyes, curly wings, and stubble bristles. Our F1 cross consisted of the F1 males we collected and true breeding virgin females with white eyes, wild type or straight wings, and wild type or long bristles. These flies produced our F2 progeny.
So now we are looking at the F1 male chromosomes for when the transgene is located on the X chromosome. Starting with the myocyte, you see that there is both an X and Y chromosome, with the red transgene located on the X chromosome. The male also has heterozygous alleles for curly wing on the second chromosome and heterozygous alleles for stubble bristle on the third chromosome. After meiosis, we see that there are 8 possible gametes formed from this myocyte. They consist of 4 sperm cells with the Y chromosome and 4 with the X chromosome. The transgene is only transmitted through the X chromosome, and it is marked by the red marker. Because of this, only the offspring that receive the X chromosome will receive the transgene. Each of the 8 cells also has both the second and third chromosome. Each sperm cell either has the wild type allele for curly wing or the dominant mutant allele marked by yellow. Each cell also has either the wild type allele for stubble bristle or the dominant mutant allele marked by the color white. Each of the 4 combinations of the curly wing and stubble bristles genes, wild or mutant, is possible. The cell could have the alleles for curly wing and stubble bristle, curly wing and wild type bristle, wild type wing and stubble bristle, or wild type wing and wild type bristle. Now we are looking at the F1 male chromosomes for when the transgene is located on the second chromosome. Starting with the myocyte, you see that there is, again, both an X and Y chromosome. In this case, the red transgene is located on the second chromosome, and the mutant allele for curly wing is also located on the second chromosome. The male is heterozygous for stubble bristle on the third chromosome. After meiosis, we observe these 8 possible gametes from this myocyte. The transgene is located on the blue second chromosome, and during meiosis is split up so that the transgene is never in the same cell as the dominant allele for curly wing, indicated by the color yellow. Each gamete also has either the dominant mutant allele for stubble bristle or the wild type allele for stubble bristle, which is located on the third chromosome. Half of the cells that have the X chromosome will either have the transgene and stubble bristle, the transgene and wild type bristle, curly wing and stubble bristle, or curly wing and wild type bristle. The same pairings of alleles apply to the 4 cells with the Y chromosome. Now we are observing the F1 male chromosomes for when the transgene is located on the third chromosome. Again, we start with a myocyte with the black X and Y chromosomes. The blue second chromosome has the mutant curly wing allele, marked by the color yellow, as well as the wild type allele for curly wing, making this male heterozygous for this trait. In this case, the red transgene is located on the green third chromosome, so the myocyte has the red transgene and the dominant mutant allele for stubble bristle on this chromosome. After meiosis, there are 8 possible sperm cells. 4 still have the X chromosome and 4 still have the Y chromosome. Each gamete includes either the allele for curly wing, marked by the color yellow, or the allele for the wild-type wing from the second chromosome. During meiosis, the third chromosome is split so that the transgene is separate from the mutant allele for stubble bristle. Because of this, half of the cells receive the transgene, while the other half receives the allele for stubble bristle. These 2 alleles are never in the same cell. The 4 cells that have the X chromosome will either have curly wing and stubble bristle, curly wing and the transgene, wild-type wing and stubble bristle, or wild-type wing and the transgene. These pairings of alleles also apply to the 4 cells that have the Y chromosome. In the case of the transgene being located on the X chromosome, the predicted offspring includes males with wide eyes and either curly wing and stubble bristle, curly wing and long bristle, straight wing and stubble bristle, or straight wing and long bristle. The expected females would have red eyes with either straight wing and long bristles, straight wing and stubble bristle, curly wing and long bristle, or curly wing and stubble bristle. Males with red eyes and females with wide eyes are not expected. For the case in which the transgene is located on the second chromosome, the predicted offspring includes males and females with wide eyes and either curly wing and stubble bristle or curly wing and long bristle. Other expected offspring would be males and females with red eyes and either straight wing and stubble bristle or straight wing and long bristle. Offspring with red eyes and curly wing and wide eyes and straight wing are unexpected. When the transgene is on the third chromosome, the expected offspring would be males and females with red eyes and either long bristle and curly wing or long bristle and straight wing. Other expected offspring would be males and females with wide eyes and either stubble bristle and curly wing or stubble bristle and straight wing. Offspring with red eyes, stubble bristle, and wide eyes and long bristle are not expected.
With 64 flies for the A3 cross in a ratio of 1 to 1 to 1 to 1, that means we would expect 16 flies for each type. For our group data, when the transgene is on the X chromosome, the p-value would be less than 0.0001, and with the independence test Yates equals 60. For the class data, when the transgene is on the X chromosome, p is less than 0.0001, and the independence test is Yates equals 1,720. When the transgene is on the second chromosome, p equals 1, and the independence test is Yates equals 0. For the class data, when the transgene is on the second chromosome, p equals 1, and the independence test Yates equals 0. When the transgene is on the third chromosome, p is less than 0.0001, and the independence test is Yates equals 21.74. For the class data, when the transgene is on the third chromosome, p equals 0.33747, and for the independence test, Yates equals 0.92.
The data gathered by our group indicates that the most likely location for the transgene is on the X chromosome. The p-value gathered for the X chromosome was less than 0.0001, which indicates a chi-square value of less than 5%, and this means the data is significant and the null hypothesis that the transgene is not located on the X chromosome can be rejected. The class data also yielded a p-value of less than 0.0001 for the X chromosome, so the statistics are in agreement. For the second chromosome, the calculated p-values for our group and the class as a whole were both 1, and the Yates value was 0. This indicated that the data exhibited phenotypic ratios very close to the predicted 1 to 1 to 1 to 1 ratio of the null hypothesis, and the transgene is not located on the second chromosome. For the third chromosome, we had a calculated p-value of less than 0.0001, while the class had a value of approximately 0.34. Based on the numbers of each phenotype we counted in our group, We concluded that it is very likely that this discrepancy could be due to error in the scoring of the progeny. We concluded this based on the fact that the number of flies with white eyes and long bristles was calculated to be double the expected number, and 0 progeny were scored with white eyes and stubble bristles. If the transgene were located on the 3rd chromosome, it would be impossible for a fly to have white eyes with long bristles or red eyes with stubble bristles. We have scored several flies with red eyes and stubble bristles, so this cannot be the case. Due to the fact that the data for the X chromosome shows no cases of white-eyed females or red-eyed males, and the chi-square values of both the class and group indicate that the expected phenotypic ratio for males and females with white eyes and pigmented eyes for the null hypothesis was not observed, the X chromosome is the most likely location for the transgene.